This is Salma Schimmel in Milan, Italy, where we continue our discussion from the European Society for Medical Oncology meeting, ESMO. And now we're going to get a recap about the findings and the data and some of the exciting news that's being presented here in the area of lung cancer. We're joined by Professor Giorgio Scagliotti, who comes to us from the University of Torino, right here in Italy.
Yes.
How convenient.
Welcome in Italy.
It's very good to see you again. You are truly one of the world key opinion leaders in the area of lung cancer. So thank you for making time to talk to us. We were at the ASCO meeting, and there we spent time with so many physicians. There was great data coming out of that meeting, some very exciting studies. But now we want to complement what we talked about at the ASCO meeting with some of the new findings and what you find the most important studies that are being presented here at the ESMO meeting.
Well, Selma, this is really an exciting time for people who are working around lung cancer as a clinician, as basic researchers. Because you was at ASCO, you heard about a new targeted agent that is called crizotinib, that is targeting a specific gene translocation, that is the ALT translocation. And in ASCO, we saw how much effective is this agent that is targeting these tumors harboring this specific translocation that is just the translocation of one gene over another chromosome. But we are now here in Milano attending the ESMO, and as you said, we have new information coming out that are following that story of the efficacy of targeted therapy in specific subgroup of patients. So over the last, let's say, 10 years, we learned that, and also from the clinical point of view and the epidemiological point of view, we discovered that there is a special subgroup of patients with lung cancer. That is the lung cancer in never smokers. So the lung cancer in never smokers is a completely different disease from the clinical point of view, but also from the biological point of view. It's a matter of fact that the vast majority of these genomic or genetic alterations are observed in never smokers, so in subjects or in patients that were not addicted by tobacco smoking. So the real reasons why we are getting this disease in never smoking is still largely unknown. I'm saying from the environmental factor or from any factor that potentially may be the cause of lung cancer in never smokers. But coming back to the genetic alteration, now in the last 4 or 5 years we got huge evidence through clinical trials, and this is telling you again how much important are clinical trials and the participation of the patients to clinical trial, because we know now that the gefitinib and the erlotinib, that are two EGFR TKIs, small molecules that are targeting the EGFR receptor, are especially active and effective in patients with tumor harboring this gene mutation in the EGFR gene. And we got in 2010 a couple of publications that were reporting on studies specifically designed in patients with tumors harboring this specific mutation in 2 specific exons of the gene, the exon 18, the exon 21. In these two studies, we detect an unbelievable activity of one single pill administered orally instead of administering chemotherapy. You know, chemotherapy is effective, but is also associated with serious adverse events sometimes. And in this case, at least the period of time for which the patients were free of disease was almost double. So let's say the possibility to get a progression was reduced of 50%. That was especially seen in the long-term follow-up. So the progression-free survival was much better with these, let's say, pills in comparison to those who were treated with chemotherapy. And because of those studies, and also because of the IPA study that Dr. Tony Mock presented for the first time 2 years ago at this meeting, we know now that the preferred treatment for patients with tumors with EGFR mutation is represented by erlotinib or gefitinib. This meeting is also important because for the first time from a Chinese cooperative group, a similar study in patients that they were defined according to the genotype. The investigators allocated patients to erlotinib or chemotherapy. The previous studies were dealing only with the comparison of gefitinib versus chemotherapy. So now we know that more or less the two agents are equally effective in the EGFR mutants. What else? Well, it is true that the patients with EGFR mutation are highly responsive to these pills, but sooner or later they will relapse. They will relapse, and obviously, again, in the last few years, we were able to understand at the biological level which are the mechanism or the biological mechanism or the molecular mechanism that are associated with this kind of acquired resistance. Now, it was found that in one of the exons of the gene, specifically in the exon 20, from the beginning, or probably as a consequence of a selection of clones, they are emerging acquired mutations that are technically called T790M mutation that are associated with the resistance to gefitinib and erlotinib. At this meeting in the presidential symposium, it was reported that one new drug from Boehringer Ingelheim that is called afatinib, that preclinically is able to inhibit not only the EGFR receptor but also the HER2 receptor that is extremely important, as you know, in breast cancer. So this drug is able to inhibit these two receptors, but more importantly, it's able to eliminate the resistance to this specific T790 mutation that is associated with the acquired resistance to gefitinib and erlotinib. So the study was a relatively large study. In the experimental arm with this new agent that is also called BIBW2992, the responses were much higher for afatinib versus placebo. Obviously, you are not expecting so much response with placebo. And the median time to progression was 3 times more in the active treatment compared to placebo. If you like to listen from me, the numbers is 3.3 months versus 1.1 month. And disappointingly, the primary endpoint was not met because the investigators were aiming to look to an improvement in overall survival. But you know, you need to look carefully to the data because we are dealing with highly selected patient population. This patient population is sensitive to many things. It's sensitive to erlotinib and gefitinib, but it's also sensitive to
we're
chemotherapy.
Talking about Tarceva and
Tarceva and Iressa, but they are also chemosensitive, or they are sensitive to the traditional chemotherapy. So an extremely high proportion of patients, in the order of 65–70% of the patients, they receive chemotherapy after the end of the study. And probably this was the reason why the investigators, they were not able to see any difference in the overall survival, because obviously chemotherapy was administered in the placebo arm, but also in the experimental arm. But when you are looking to the absolute numbers, you are ending up in a median survival time that is almost 21 months. That in stage 4 disease is really unusual to be seen. Now, all these data, they need to be carefully considered even when you are dealing with negative data. Because obviously, if you are a never smoker, you have the possibility to have an extension of your life of 2 years is in any way relevant. In addition to what I was mentioning you just now, there were also other 2 studies reporting on a potential other mechanism of resistance, that is the c-Met amplification. This is another mechanism of resistance that has been detected in tumors that became refractory to Tarceva and IRS. Sorry. As you know, c-Met is a molecular pathway that is associated with the possibility of malignant cell to disseminate to distant sites. It's not only related to the possibility to generate metastasis, but also to the possibility to invade the normal tissue, to proliferate locally. In a couple of studies, it was detected that using a monoclonal antibody against the cellular receptor for the c-Met pathway, and the ligand of this receptor is the hepatocyte growth factor. And in another study, the use of another small molecule that is targeting the internal domain, so the part of the receptor that is lying internally to the cell, it was found in the 2 studies that these agents may be useful in reversing in some way the resistance. And consequently, we can envision a future scenario where, in addition to erlotinib, you can combine one of these 2 agents. And for one of these 2 agents, for the monoclonal antibody that is called MET-MAB, they found also a biomarker. And the biomarker was the possibility to detect in the tumoral tissue a positivity at the immunohistochemistry level for c-Met. If you are positive for c-Met, you have much more chances to get a benefit from this monoclonal antibody. In the other study, they were following a different approach. They were trying to compare, in a phase 2 randomized clinical study, the possibility to administer erlotinib alone as a second-line treatment, or erlotinib plus RQ197, that is this small molecule that I mentioned before. That was just a 200-patient study, but it was very well designed. It was designed in a way to look broadly to the possibility to look to the different subgroup of patients. They found that the combination of RQ197 plus erlotinib is more effective in non-squamous histology. But they were also smart because they were collecting tissue. And you know, tissue is the issue in terms of targeted therapy, because you need to detect the target, otherwise your targeted therapy is not effective.